Welcome, everyone. My name is Jessica Blaustein. I'm the Senior Account Director of the Healthcare Partnerships team at the Muscular Dystrophy Association. Thank you for joining us today for the MDA Industry Update webinar and for all that you do for MDA and the neuromuscular community. This webinar, titled Introducing INS1201 and the ASCEND Study, Gene Therapy for Duchenne Muscular Dystrophy, is sponsored by Insmed. MDA's industry update webinars provide an opportunity for the medical community to receive information and updates impacting care and treatment directly from pharmaceutical and biotechnology companies. These webinars are just a part of MDA's medical education resources. We encourage you to visit the medical education pages of mda.org, where you'll find resources including MDA's Clinical Research Awareness Program, Grand Rounds webinars, our MDA Monthly Report e-newsletter, and much more. We'll conclude this webinar with a brief Q&A. Please put your questions into the Q&A as they arise, and the speakers will address them if time permits at the end. Now, I would like to introduce our presenters for today's webinar. We will hear from Dr. Aravindan Veerapandiyan, Dr. Panda, as he is more commonly known, MD, pediatric neurologist and director, Comprehensive Neuromuscular Program, and co-director, Muscular Dystrophy Association Care Center at the Arkansas Children's Hospital, and Dr. Mark Stahl, MD, PhD, Executive Director, Clinical Development at Insmed. And now, without further ado, I'll turn it over to Stephanie Duffy, Head of U.S. Patient and HCP Advocacy and Engagement, Gene Therapy, for an introduction to Insmed. Thank you, Stephanie.
Thank you, Jessica, and the MDA team, and hello to everyone. Thanks for taking some time out of your day today to be with us. We greatly appreciate it. And introducing everyone to Insmed, we think it's important to highlight the mission of our work here and highlight that it's done alongside all of you. Over the past few years, we've had the opportunity to speak with and collaborate with folks like Dr. Panda and other physicians within the community, as well as our patient advocacy partners, caregivers, and those at the leadership team of MDA. So our work is certainly not done in isolation, and while we have a very tenured and talented team, the clinical development program, our protocol, and all of the work that we're bringing forward has been alongside all of you. So thank you in advance for that support, and we're excited to talk a bit more today about our preclinical work as well as our first in-human trial in Duchenne Muscular Dystrophy. Jessica, thank you for the introduction. One thing I'll add is that while I work in advocacy at Insmed, I also work in advocacy at home. I am a mommy of 3. My little one lives with a rare neuromuscular condition, so this feels like the most relevant place for my professional work here. And I'm delighted to be alongside all of you today for this. And so with that, I will pass it along to my colleague, but my friend, Mark Stahl, to get us started. And also thank you, Dr. Panda, for being with us today.
All right. Well, thank you, Steph, and also Jessica for the introductions. As we heard, my name is Mark Stahl, and I work here at Insmed in clinical development, and I'm myself a neurologist and scientist. It's my pleasure to get to talk about sort of the rationale and the genesis of this exciting project. First, let me introduce INS1201. For those of you who aren't familiar with our program, INS1201 is a microdystrophin-based gene therapy that is delivered via AAV9. We're studying it for its potential to replace the missing dystrophin protein in boys with muscular dystrophy. The delivery, the administration of this drug is really one of the interesting features of it. It is given as a one-time intrathecal injection of a fixed dose into the CSF, which is, of course, in contrast to how most of the currently given gene therapies for Duchenne muscular dystrophy are given. And I think that's maybe the part of the program that brings up the most questions, and so we'll spend a little bit of time talking about exactly why we think that's a good idea and how that seems to work. In terms of the actual drug itself, you'll see in the diagram above, you'll see the AAV9 is the capsid, and then the microdystrophin construct contains exons 1 through 17 and 59 through 69 of the dystrophin gene, as well as uh, under the control of the MHCK7 promoter. And I think a lot of characterization has already been done of a lot of these parts, but we are the first to put them together in this way and deliver them in the way that we are. So let me move right into where this idea of giving a muscle-directed gene therapy into the intrathecal space sort of came from. The observation that this was possible was really made and published by people who are now at Insmed, as well as others, who were really initially looking at AAV9 to deliver genes to the CNS and not so much to the muscle. But in those studies of biodistribution, clearly high levels of transduction in skeletal and cardiac muscle were observed when the animals were looked at in their studies. And alongside that, comparatively low levels of expression and transduction were seen in other peripheral tissues, and most notably the liver. And so this was part of the genesis of the idea that one could intentionally inject AAV into the CSF, avoid first-pass liver effects and high levels of immune activation, and result in efficient delivery to the muscle. These preclinical studies in non-human primates were then more formalized and taken directly into comparison studies. And you can see one of those studies. The results are here on your screen. You can see on the very top is some non-injected control, and then right below that we demonstrate some intravenous injections at 2 different dose levels, and then below that, for comparison, we have AAV9 at 3 different dose levels, the top 2 of which match the exact same doses given intravenously. This is a marker. We just used GFP here just to show the distribution and transduction, but you can see just visually, even without quantifying it, that the delivery of AAV to the muscle and the resulting expression of the marker gene is actually very high in the muscle when given intrathecally as compared to the intravenous route. And then if you look over onto the far right, you can see the 2 panels marked heart and liver. You can see the cardiac expression in these animals is very high, but notably the liver expression was comparatively low, and this really is a big part of the genesis of why we think this route of administration is both efficient and potentially quite safe, and that's what we did end up seeing in our preclinical studies that have been done so far to date. So of course, once that observation was made for the use of this in DMD, we then wanted to confirm naturally that we could do this with dystrophin in our old friend, the MDX mouse. And you can see on the left panel the effect that increasing doses have on both muscle architecture and on dystrophin expression. As a dose-dependent effect is demonstrated with the collagen infiltration into the muscle, as well as the percentage of dystrophin positive muscle fibers. And then on the right, you can see, of course, that along with the appropriate, histological improvement, we're also able to see functional improvements in muscle. And so we measured both the total amount of force that the muscle is able to produce, as well as a functional test of forelimb strength. And you can see grip strength. And you can see this here. These were taken at about 9 months after the administration of INS 1201. We then continued some of these studies out for durability. And you can see here that after 1 year, and some of these studies were done even later than that, there is a persistence of effect of the dystrophin expression and really maintained positive effects on the muscle architecture as well at the 1 year time point. In addition to the skeletal muscle benefits, we of course also look at heart and cardiac muscle. Much as what we saw in our initial observations in the non-human primate, we do see really a very robust distribution of INS1201 to the heart once we deliver it into the intrathecal space in these MDX mice. So along with that, of course, comes evaluation of safety. We performed toxicity studies in both the mouse and the monkey. Overall, INS1201 was very well tolerated without any observed adverse effects. The highest dose level that was tested in both of these studies was actually the maximum feasible dose that we could give to the animals. That highest dose scales to about 33% higher than the maximum dose we're planning for our clinical study. The tolerability in animals, of course, based on observations of behavior and signs of distress, but then also, of course, we follow changes in laboratory values that were also provided a positive result in terms of tolerability and safety. We had only transient changes in some laboratory values in isolated individuals. We performed electrodiagnostic studies to evaluate for any potential effects on the dorsal root ganglia or sensory systems, and nerve conduction studies as well as EMG, and we had no abnormalities there. And the histopathology was performed on multiple tissues, including the DRGs, with no concerning pathological findings. In addition to that, we of course use those studies as well to observe for consistency in the biodistribution, and so we were able to develop essentially a dose equivalent or translation from mouse to monkey that allowed us to develop the appropriate dose levels for a first-in-human study that will be a fixed dose into the CSF space. And so with the dosing is essentially based on the concentration of virus that we see in the CSF. So a little bit just to kind of summarize those preclinical results. Intrathecal introduction of INS1201 results in high levels of microdystrophin expression very efficiently with low levels of impact on the non-muscle tissues. Improvements in the muscle function and tissue pathology. The expression of dystrophin is maintained at about 1 year and appears to be quite durable. And the toxicology studies demonstrated that the treatment was well tolerated with no observed adverse events at the doses we studied. And so you can hear on the right, this is the result of those animal studies in terms of their influence on our study design. We'll be using two doses that both of which were identified in translation studies from our mice and non-human primates, and you can see those on the right. We'll talk a little bit more about those in the next part of the talk. One other point that I want to bring up in terms of our preclinical work really addresses the efforts that Insmed has put into optimizing the manufacturing analytics to make a product that is not just consistent, but also very high purity. And so we focused quite a bit of our effort into reducing the amount of empty capsids, as well as having a high quality, consistent drug from batch to batch. And so with that said, I think although there's always more to say, I think I'll pause here and move the talk over to Dr. Veerapanian to talk a little bit more about our first-in-human clinical study that he and his team have been participating in, and let him give his overview of our study.
So thank you, Mark.
All right.
All right. I get to do the easy part of it. So, all right. I just wanted a reminder, please feel free to post your questions in the Q&A tab, and we'll try to get them all answered at the end. The study that we're going to talk about is the ASCEND study. It's a phase 1 safety study. It's a multi-center single-arm phase 1 study enrolling boys with Duchenne muscular dystrophy that are 2, 3, and 4-year-old. And as Mark kind of said, we're looking at 2 dose levels here. And the primary objective was to evaluate the safety and the tolerability of a single dose of intrathecal INS1201. The secondary objectives, of course, based on these two, how kids do with these 2 dose levels, we would identify a single dose for the efficacy study moving forward, and also looking at the microdystrophin expression from the muscle biopsy. And there are also other functional assessments that's being done in the study that includes North Star Ambulatory Assessment, their stride velocity, P-body in the younger age group, as well as GROW, and some of the time function tests as listed here. And some of these assessments are only applicable for kids that are 3 and 4-year-olds, such as North Star Ambulatory Assessment, since some of the younger kids may not be able to do those. All right, next slide. This kind of gives you the enrollment schematic here. So the ASCEND has 2 parts, like part 1 and part 2. Part 1 includes 3 and 4-year-olds, and part 2 includes 2 and less than 3-year-olds, 2 to less than 3-year-olds. And as you could see here, the first, the starting dose is the first dose 5 into 10 to the power of 14 vectograms, the single fixed dose. And once you have the patients enrolled in that cohort, you'll have IDMC by Safety Independent Data Monitoring Board review on the safety. And then, based on the review, and they'll let us permit to move on to the next cohort. And that's how each cohort would play. And I think to date, we have cohort 1 that has been fully enrolled. I also want to comment that IDMC typically meet within 30 days after the completion of 30-day safety period of the 3rd patient enrolled into each cohort. All right, next slide. To kind of touch upon the inclusion-exclusion criteria, it's you have to have Duchenne and age less than 2 to less than 5 years old, as we discussed before, and they have to walk unassisted for at least 10 meters, and definitely diagnosis of Duchenne muscular dystrophy. And the genetic mutation is something I want to highlight here. So we're including only patients that have mutations in between exons 18 and 58, including 18 and 58. So the ones before exon 18 and after exon 58 would be excluded, irrespective of what type of mutation that they have. And the ejection fraction has to be 50% or higher. And the AAV9 antibody titers has to be less than 1 to 50, as determined by ELISA. Next slide. Mark, can you hear me? Stephanie, Mark might be, he
Yep.
looks frozen on my screen.
Mark looks frozen on my screen too, so he may be redialing back in. So I'm sure he'll be back with us shortly. But the good news is we do have another member of our clinical development team on, as well as medical affairs.
All right. Do you want me to pull up the slide? I can and start sharing.
Sure. Oh, that's right. It was being shared from Mark's laptop. Sure.
Sorry, guys.
That's okay. If that's convenient for you, certainly you can pull them up and continue navigating through.
Yeah.
Appreciate you doing that. While you're pulling that up, Dr. Panda, we're noticing some questions coming in through the Q&A, so it might be worth just addressing it as we go through here, if that works for you. And one of the
Sure.
questions that has come up a couple times is just further inquiry, and this could be for our clinical development team as well as yourself, on why the selection around the specific age group for this particular study. So this slide actually may be a perfect place to land on that question that has come in from a couple of folks attending today.
Uh, uh, Dr. Panda, if you feel you can go first and I can kind of compliment you.
No, you go for it for one.
All right. One of the things that, thank you, Dr. Panda. So one of the things that we wanted to concentrate on is the the age group that you're looking at on the screen is it's not been investigated in other clinical studies. So first in human studies, we wanted to look at it. And the other thing is that the animal studies that we have done is how you actually what's called the translational research spectrum. So we translated the work that we did in animal studies, including non-human primates, and the scale up that we did. So we carefully kind of translated into this age group based on the CSF space. So it's a IT injection. In this study, we have a IT injection versus IV. So we did that translation in the animal studies. We scaled it up to this age group based on the CSF space in this age group. So those two factors, the animal studies and how we translated that information or data into first in human studies, and the fact that this age group has not been investigated in other clinical studies was the motivation or the drive for us to pursue this age group.
And I hope as how we do with this, there might be plans in the future to expand to other age groups as well.
Great. All right. Thanks for pulling those slides back up, and we can certainly keep going.
All righty. Sorry, guys, for that interruption. And I think this kind of tells you the follow-up or the study periods. It has four study periods. You have the screening or pre-treatment period, which is up to 46 days, and that's when all of your screening activities look for if as you know, if the kid would be eligible, and the peri-dosing, peri-infusion steroids start on day -1. Then you have your intrathecal injection of INS1201 happening on the treatment day, and it's most of the times either sedation or anesthesia. And a lot of times what people do is they try to combine that along with biopsy. And we also get nerve conduction studies in the study to given the possibility of DRG toxicity in the intrathecal injection. So that also happens on the same day. We try to coordinate those, and they stay overnight for monitoring. And after that, they're followed up for up to 100 weeks, and that's your safety monitoring period and also follow-up periods. And they get needle biopsies at 16 weeks, 48 weeks, and optionally at 96 weeks. And there's also as some of the safety blood work was there's home health option to do that as well. And then the post-dose prophylactic steroid has been it's there for 12 weeks, and then they'll come off and they go back to their regular standard of care steroids, or if they are not on steroids, then they go back to not on steroids, and then depending on the kid's clinical picture, if you need to have them on a standard of care steroids, they'll go back on that. And after that, there's a separate extension follow-up studies that's for 3 years as well. And there is, as any other study, there's an oversight by an independent data monitoring committee. And like I think we don't have to talk about this much, but there are study-related support available, like that we have for most of the studies. We don't want the distance or any other things to be a barrier for the participation. So there are vendors that help with your logistics, prepaid services, expense reimbursements, and all of that. So these are the sites that are currently planned for the study, and the ones that has the yellow stars are the ones that are open and enrolling. And I think there's a couple more that will be activated in the near future. And you can go to clinicaltrials.gov, obviously, to find more information. And there's also a website for the study itself, which you could refer to, and also contact information for Insmed here. I think that's all I had in summary. Just to kind of sum up what you heard from Mark. So in the preclinical animal models, the treatment with INS1201 yielded microdystrophin expression in both animals studied. And also you saw some improvement in the muscle function in the mouse model with DMD. The distribution to skeletal and cardiac muscle, along with low expression or low distribution in the liver of the animal, suggests that intrathecal delivery may be warranted further study. And there were no observed side effects that were seen at the highest dose tested in the animal toxicology studies. Together, their preclinical data supported the approach for the clinical trial, the phase 1 first in human that we talked about. Ascend, it is underway. This is the first in human study that is evaluating the safety and tolerability of a single intrathecal dose of INS1201. And the study also evaluates what is the recommended dose for future efficacy studies. As you heard, they're looking at 2 different doses, and the age group is 2, 3, and 4-year-olds with Duchenne that are ambulatory. With that, I thank you guys for listening, and we'll get into Q&A with Stephanie.
Great. Thank you, Dr. Panda. And we do have Mark back as well. Mark's computer crashed, of course, right? But
Sorry, everyone.
No worries. These things happen, and appreciate you being able to jump back on. So thank you both. One question that has come up from several different folks attending is just if we can just revisit, Mark, and for Quan and Dr. Panda, just the rationale of intrathecal route of administration and why that's been selected. I think some folks just wanted to hear that perhaps one more time just for additional clarification.
Sure. As I said, it really began as an empirical observation that when AAV9 in particular was given intrathecally, that the biodistribution that resulted included a very high degree of delivery to skeletal and cardiac muscle, and that on top of that, the delivery to liver was lower. The reasons for that are, I will say, currently under investigation. We don't fully understand it. There are a few reasons why that might be. One is that, of course, the initial administration of high doses of virus may activate the immune surveillance cells in the liver very quickly, causing a trapping of large quantities of virus in the liver, over the first few hours. Whereas what we see in our animal studies is that while we do have virus that ends up in the blood, in the circulatory system, the release is much slower over the course of several days. And that difference alone may account for some of the difference there. There are some other reasons why delivery to the muscle may be particularly efficient, and that may include the sort of anterograde transport directly along neurons to the target tissues, which include the muscle, and some work is ongoing to sort of look at exactly how much of a contribution that may be. And so there are a few other potential mechanisms as well. But at the end of the day, that is actually still an ongoing area of investigation.
Great. Thanks, Mark. I recognize we are right at that half-hour mark. So thank you for the contributions of this team today. And as Jessica mentioned in the upfront, This webinar has been recorded and will be available via MDA and also to all the centers of excellence for the ongoing education. As mentioned as well prior, you can certainly visit Ascend or Ascend DMD website just to learn more about clinical trial sites and more about the trial to come. And we look forward to seeing everyone in the new year at the upcoming scientific meeting and continuing to further partner with all of you. So thanks again to the MDA team and wishing all of you a wonderful holiday season. We'll look forward to catching up soon. Thanks, everyone.
Yes.
Bye-bye.